am Gianluca Vella. I'm a postdoc at the Humanitas Research Hospital, and I'm part of FIBROTARGET Consortium. In this section, I will be talking about how to employ ex vivo human intestinal explants to study fibrosis. I will be focusing on two different approaches, the biopsy and the ex vivo organ culture, EVOC, both starting from transmural resection of human intestine. Starting from the biopsy, first of all, the intestinal resection is collected at the surgical room, and then in the laboratory, the tissue is washed and all the mesenteric material is removed. Then the transmural biopsy is taken using a punch biopsy device, and the biopsy is cultured. This is the general experimental design that is used for culturing the biopsy. At time zero, the stimulus or the vehicle is administered, and the tissue is incubated for 16 hours at 37 degrees, 5% CO2. At the end point, the supernatant and the tissue are collected for downstream analysis. This is the list of all the possible stimuli that can be had in the medium, inflammatory mediators, drugs, metabolites, and also bacteria. In the case where the surgical resection biopsies are not available, endoscopic biopsies could be used instead in this protocol. So the supernatant can be used to analyze the modulation of profibrotic cytokines, including PDGF, TGF-beta, TNF upon treatment, while the snap frozen tissue is used to assess the modulation of profibrotic genes, including alpha-SMA, CTGF, and FAP. The EVOC is a more complex system. It's a polarized system. The protocol for the culturing of EVOC, starting from human intestinal mucosa, has been published many years ago from the laboratory of Maria Rescigno. Similarly to the biopsy, the intestinal section is collected at the surgical room, and then the laboratory, the tissue is prepared, and then it's ready to be used for the EVOC mounting. On the contrary of the biopsy, where the tissue is completely submerged in the medium that contain the stimulus, in the EVOC system, the intestinal tissue lays on a grid that is above a petri dish that contains the medium. The intestinal tissue has the mucosa surface facing upwards, and a plastic cylinder will be glued on the mucosal surface. And this is where the stimulus will be added. Also, in this case, the stimulus can be an inflammatory mediator, drug, metabolites, bacteria, and so on. This is the general experimental design that is normally used for the EVOC system. The EVOC is incubated for 1 hour at 37 degrees to allow mucus reconstitution, and then the stimulus or the vehicle is added. The EVOC then is placed on an aerobic jar that is filled with 100% oxygen at 1 atmosphere and is incubated at 37 degrees for 4 to 16 hours. At the end point, the subnatant that contains all the factors that were released by the tissue upon the treatment, and the tissue per se is collected for downstream analysis. Some key recommendations are to keep the apical volume minimal to preserve the oxygen diffusion, but also to limit the culture duration to maximum 16 hours to avoid the tissue damage. This is the list of all the possible readouts for this system. The tissue that is collected can be used for histology. For example, it will be possible to do H&E staining, Alchampas, or Sirius red staining. The tissue can be also stained for immunofluorescence to assess some general markers of intestinal barrier integrity, including MUC2, ZO1, PP1, CD31, or can be stained for some specific markers of fibrosis, including CD34, FAP, collagen 1, twist 1, apodoplanin. The tissue collected can be also analyzed to assess the modulation of profibrotic genes upon the treatment, including CTGF, FAP, alpha-SMA, and subnatant can be analyzed for assessing the modulation of the profibrotic cytokines, such as PDGF, TGF-beta, TNF. These are some representative histological pictures of EVOC that have been treated at different concentration of LPS. And in this example, by the H&E staining and the ultrasound staining, you can appreciate how we can indeed monitor how the architecture and the mucus is modulated, is altered by the LPS administration at a dose-dependent manner. To summarize, both biopsy culture and EVOC are successful ex vivo approaches to study fibrosis. In general, the biopsy culture is recommended when the starting material to begin with is very limited. However, it does not allow for polarized stimulus administration or histological analysis. On the contrary, the EVOC allows for polarized stimulus administration and histological analysis. However, it requires specialized equipment, for example, the aerobic jar.